Slime is everywhere, on rocks in streams, on the underside of leaves, in tidal pools, inside your own body. It's one of the most common substances on Earth. And yet, most people never think about it, never consider what it is, never appreciate the quiet physics that makes slime what it is. But tonight, we're going to look closer. We're going to understand this slow, patient, peaceful material that exists all around us. At its simplest, slime is a viscous fluid. That means it flows, but it flows slowly, much more slowly than water. When you pour water, it moves quickly. It splashes, it spreads, but slime moves gradually, patiently. It oozes, it creeps, it takes its time. There's no rushing slime. It moves at its own pace. And there's something deeply calming about that, about a substance that simply cannot hurry. But the science behind this is surprisingly simple. Slime is made of long molecules called polymers. These are chains, long strings of atoms connected together. In water, molecules are small and simple. They slip past each other easily. But in slime, these long polymer chains tangle together. They slide past each other, yes, but slowly. They have to untangle, have to rearrange, have to find their way through the maze of other chains. This takes time, and that's why slime flows the way it does. Slow, steady, patient. If you've enjoyed learning about the peaceful science of slime, if this journey into slow-moving substances brings you calm, consider subscribing to Simple Science for Sleep. We explore topics like this every night. Things that seem simple but hold surprising depth. Things that help you relax and learn at the same time. Please take a moment to like this video and leave a comment below. Let us know where you're watching from and what time it is there. Are you already in bed, sitting somewhere quiet? Wherever you are, you're part of this community. People all over the world finding peace in unexpected science, finding rest in understanding. The viscosity of slime can vary enormously. Some slime barely flows at all, sits in a blob, moves only when forced. Other slime flows more readily, spreads slowly across surfaces, drips gradually from edges. The difference is in those polymer chains, how long they are, how tangled, how sticky. More tangling means slower flow, means more viscous slime, means more patience required for the slime to move. Temperature affects slime too. Heat makes molecules move faster, makes polymer chains slide past each other more easily. Hot slime flows faster than cold slime, not quickly. Slime is never quick, but warmer slime is less viscous, more willing to move. Cold slime becomes even more patient, even more resistant to flow. In winter, slug slime moves differently than summer slime, slower, thicker, more deliberate. Slime is what scientists call a non-Newtonian fluid. This means it doesn't follow the normal rules of liquid behavior. With water, if you stir it gently or stir it hard, it behaves the same way. It's always liquid, always flows the same. But slime changes. Some slime gets thinner when you stir it, flows more easily when disturbed. Other slime gets thicker, resists harder when you push it. The material responds to force, changes its properties based on how it's treated. And this is remarkable when you think about it. A substance that adapts, that changes its behavior based on circumstances. There's no consciousness in this, no decision, just physics, just the way polymer chains respond to stress. But it feels almost intelligent. This substance that becomes more fluid when handled gently, that resists when forced. It's as if slime knows something we often forget. That patience is more effective than force. That slow, steady movement gets you where you need to go. that there's no need to rush. Humans have been making slime for thousands of years. Ancient adhesives were often slime-based. Tree sap, plant resins, mixtures of materials that dried slowly, that held things together. Modern slime, the kind children play with, is usually made from polymers like polyvinyl alcohol, mixed with borax or other cross-linking agents. The result is that satisfying texture, that slow pour, that resistance to flow. The science is the same as natural slime. Long chains, tangling, patient movement. And this patient movement appears in the natural world in beautiful ways. Watch a slug move across the ground. Really watch it. You'll see something remarkable. It leaves behind a trail, a glistening path of slime. This isn't accidental. This isn't waste. This is how the slug moves. It creates the slime, lays it down like a road, then glides across it. The slime is both the path and the vehicle, both the surface and the lubricant. The slug is constantly producing this mucus, never stopping, always making more. The slime comes from a gland under the slug's head, the pedal gland. It secretes mucus continuously as the slug moves. This mucus is mostly water, about 95% water. But the remaining 5% is what makes it special. Glycoproteins, long molecules that create that thick, viscous texture, that patient flow. The slug controls the consistency, can make it thinner or thicker, depending on what it needs. On smooth surfaces, thin slime. On rough surfaces, thicker slime for cushioning. This slime serves multiple purposes. First, it allows movement. A slug has no legs, no feet, just a muscular foot that ripples in waves. These waves push against the ground, but skin on dirt would create friction, would make movement difficult. The slime eliminates this friction, creates a slippery surface where the slug can glide. Without slime, a slug couldn't move, would be stuck, helpless. The slime is essential, not optional, not luxury, absolute necessity. Second, the slime protects. Slugs have soft bodies, no shell like snails, no armor, just vulnerable flesh. The slime creates a barrier, keeps moisture in, keeps threats out. Rough surfaces that would scrape the slug's skin slide harmlessly over the slime layer. Bacteria and parasites have a harder time penetrating the mucus. It's defense, it's armor made of goo. Simple but effective. The slime also tastes bad, bitter, unpleasant. Predators who bite into a slug often spit it out. The mucus coats their mouth, leaves a terrible taste, makes the slug not worth eating. This is passive defense. The slug doesn't fight, doesn't run fast, can't run fast, just makes itself unpalatable, makes eating it more trouble than it's worth. And this works. Slugs survive, thrive, spread across the world using nothing but slime and patience. That silver trail a slug leaves behind, that shimmer you see in the morning on sidewalks, that's dried mucus. It catches light, reflects it, creates that pearlescent shine. Each trail is evidence of a journey, of a slug moving slowly through the night, searching for food, for moisture, for whatever slugs seek. The trail can last for days, weeks if conditions are dry, a record of movement, of a creature that was here, that passed this way, moving at its own unhurried pace. Slugs move about 1 meter per hour at their fastest, usually slower, much slower. There's no rushing a slug. They can't rush. Their biology doesn't allow it. They move through the world at the pace slime allows. And this is enough. This is sufficient. They find food, they reproduce, they survive. Speed isn't necessary. Patience works. slow, a steady movement through the darkness, leaving trails of silver, leaving evidence of their patient existence. When a slug encounters an obstacle, it doesn't go around if it can go over. The slime lets it climb vertical surfaces, lets it stick to glass, to walls, to the underside of leaves. It can hang upside down, defying gravity, held only by its slime. The mucus is sticky enough to hold its weight, but not so sticky it can't move. It's a perfect balance, a precise viscosity that allows both adhesion and motion. Too sticky, and the slug would be trapped. Too slippery, and it would fall. But the slime is exactly right, exactly what's needed. Different slugs produce different slime. Some species make particularly sticky mucus, others make more fluid slime. There are slugs that can make their slime stringy, can hang from it like a rope, lowering themselves down from high places, climbing back up the same slime thread. It's remarkable when you think about it. A material the slug produces, uses, then sometimes eats afterwards to reclaim the nutrients. Nothing wasted, everything recycled. And these are not the only creatures that rely on protective slime. Snails are slugs that learned to build homes. That's essentially what they are. Close relatives, similar bodies, similar slime. But snails carry shells. Portable shelters they never leave. And the slime plays an even more important role for them. It's not just for movement, not just for protection. It's also for sealing, for survival, for patience beyond what most creatures can manage. A snail produces mucus just like a slug does, from glands in its foot. But snail mucus is often even more remarkable. It has to work on surfaces slugs never encounter. Glass, smooth walls, wet rocks. The snail needs to climb these surfaces, needs to stick while moving. The mucus does this, creates suction, creates adhesion. A snail on glass is held there by mucus alone. No other attachment, just slime making contact, creating enough grip to hold its entire weight, shell and all. The chemistry of this is elegant. The mucus is hydrophilic, loves water. On wet surfaces, it spreads, creates a thin film. This film excludes air, creates a seal, like a suction cup but softer, more adaptable. The snail can move by lifting part of this seal, shifting its weight, creating new contact points while releasing old ones. It's a slow process, a patient process, but it works so well that snails climb vertical surfaces in rain, when everything is slippery. Yet when most creatures can't get traction, snails just keep climbing. But the most remarkable use of slime comes during drought, when conditions get dry, when moisture disappears. A snail will retreat into its shell, pull its entire body inside, and then it does something clever. It secretes a special kind of mucus, different from movement slime. This mucus is designed to dry, to harden. It forms a seal across the shell opening, a door made of dried slime. This is called an epiphragm, a protective barrier between the snail and the hostile dry world. Behind this slime door, the snail waits. It doesn't die, doesn't truly sleep. It enters a state called estivation, like hibernation, but for heat and dryness instead of cold. Its metabolism slows way down, almost stops. Heart rate drops. Breathing becomes barely perceptible. The snail uses almost no energy, almost no water. It can stay like this for months, sometimes years, just waiting, perfectly patient, perfectly still, protected by a door of dried slime. When rain finally comes, the snail knows. Moisture penetrates the shell, touches the dried mucus. The epiphragm begins to soften, to dissolve. The snail becomes active again, Metabolism increases. It breaks through the slime seal, emerges, begins moving, searching for food, for water, for everything it needs. And if conditions dry up again, it just repeats the process, seals itself in, waits. There's no stress in this, no panic, just patience, just trust in the process. Trust that rain will come eventually, and that the slime will keep it safe until then. Some desert snails can survive like this for years, 5 years, 10 years in extreme cases, and waiting, sealed behind their slime door, using almost no resources, just existing in a state of profound patience. When scientists first discovered this, they didn't believe it. Found snails in museums, glued to display boards, snails that had been dead for years. But when they added water, the snails woke up, broke through their old dried slime, started moving. They hadn't been dead, just waiting, just being patient, for years. The slime that allows this is different from movement slime. It has different proteins, different structure. It's designed to dry into something hard, something durable, but also something the snail can break through when needed. It's strong enough to keep moisture in and predators out, but weak enough that a slug's muscular foot can break it. This balance is precise, evolved over millions of years, perfected through countless droughts, countless snails that survived or didn't based on the quality of their slime seal. Young snails learn nothing about this. They just know. It's instinct. When conditions get dry, they retreat. They seal. They wait. No one taught them. No parent demonstrated. The knowledge is built into their biology, into their genes. This is how snails have survived, how they've spread across the world, not through speed, not through strength, through patience, through slime, through the ability to simply stop, to wait, to trust that conditions will improve, to let time pass while sealed safely away. The trail a snail leaves is different from a slug's trail, usually narrower, often more precise, because the snail is heavier, needs better adhesion. The mucus is slightly different in composition, slightly stickier, slightly more viscous, but it's still fundamentally the same. Long polymer chains, water, glycoproteins, simple ingredients creating complex properties, creating the ability to move, to stick, to seal, to survive. And slime creates survival in even stranger ways. There are organisms on Earth that don't fit into our normal categories. They're not plants. They can't photosynthesize. They're not animals. They don't have muscles or nerves or organs. They're not fungi exactly, though they're close. They exist in their own category, and they're made almost entirely of slime. These are slime molds, and they're among the strangest living things on the planet. A slime mold starts as individual cells, tiny amoebas living in soil or rotting wood, each one independent, each one living alone. But when food becomes scarce, and something remarkable happens, the individual cells come together. They merge, hundreds of them, thousands, sometimes millions. They form a single large organism, a blob of slime that can be bigger than your hand. This blob isn't a colony. It's not individuals working together. It's actually one organism now, one giant cell with thousands of nuclei. All those individual cells gave up their independence, became one thing, one living mass of slime. This mass can move. It doesn't have muscles, doesn't have any motor organs, but it flows. The cytoplasm inside it streams, flows from one part to another. This creates movement, slow movement, patient movement. The slime mold creeps across surfaces, searching for food, for bacteria, for yeast, for anything it can consume. It moves maybe a centimetre per hour, sometimes faster, sometimes slower. There's no rushing it. It goes where it wants, at its own pace, following chemical signals, following gradients it can sense, moving toward food, away from light, responding to its environment without any nervous system to process information. Slime molds can be beautiful, bright yellow, orange, white, pink. They look like spilled paint spreading across a log, like alien life colonizing the forest floor. But they're completely natural, completely ordinary. They exist in every forest, every place with decaying wood and moisture. Most people never see them, never look closely enough, never slow down enough to notice these remarkable organisms living quiet lives in the leaf litter. The most studied slime mold is Physarum polycephalum. It's yellow, it's easy to grow, and it's smart. Not smart like an animal. It has no brain, no neurons, no nervous system at all. But it solves problems, it makes decisions, it learns. Scientists have tested it in ways that reveal its unexpected abilities, and what they've found is remarkable. This brainless blob of slime can do things that seem impossible for something so simple. Put a slime mold in a maze with food at the end. It will explore the maze, send out tendrils in every direction. These tendrils branch and spread. Some reach dead ends. Those tendrils eventually withdraw, retreat. The slime mold pulls itself back, tries other paths. Eventually, a tendril finds the food, and then something remarkable happens. The slime mold retracts all the wrong paths, all the dead ends, all the inefficient routes. It keeps only the shortest path from start to food, the most efficient route, the solution to the maze. It does this without thinking, without planning, without any mental process at all. It does it through chemistry, through feedback. Tendrils that find food send signals, chemical signals that strengthen those pathways, make them thicker, make more cytoplasm flow through them. Tendrils that don't find food eventually starve, thin out, disappear. What remains is the solution, the answer, the best path. The slime mold has solved the maze through simple rules, through chemical gradients, through patience. This takes hours, sometimes days. The slime mold doesn't rush, can't rush. It explores methodically, thoroughly. Every path gets tested, every option gets tried, and gradually, through this patient exploration, the answer emerges. Not through intelligence as we understand it, but through a different kind of problem solving. Distributed, or chemical, efficient. The slime mold knows nothing about mazes, about efficiency, about optimal paths. It just follows simple rules. And those rules lead to complex solutions. Scientists have tested slime molds with increasingly complex problems. Multiple food sources, obstacles, harmful substances to avoid, and the slime molds solve them all. They find configurations that minimize distance, that avoid dangers, that efficiently connect all food sources. They create networks, branching patterns, structures that look designed, that look intentional. But they're not. They're emergent. They arise from simple rules played out patiently over time. What's remarkable is the patience in this. A slime mold doesn't get frustrated, doesn't give up, doesn't rush to find an answer. It just keeps exploring, keeps flowing, keeps trying paths until something works. There's no anxiety in it, no stress, just steady, patient problem solving through simple chemistry, through the slow flow of slime from one place to another, through giving every option enough time to prove itself useful or not. And this patience, this simple approach, leads to solutions that seem remarkably intelligent. How does a slime mold know where to go? How does it decide which path to take? How does it solve problems? These seem like questions that require a brain, require thought, require consciousness. But a slime mold has none of these. It has no central processing, no command center, no decision-making organ. It's just slime, just cytoplasm in a membrane. And yet, it behaves as if it's thinking, as if it's making choices, as if it knows what it's doing. The answer is chemistry. Simple chemistry happening throughout the organism. Every part of the slime mold can sense its environment, can detect chemical gradients. Food releases certain chemicals. The slime mold's membrane has receptors that detect these. Where the concentration is higher, that part of the membrane becomes more active, more permeable, more likely to allow the cytoplasm to flow in that direction. It's not a decision, it's just chemistry, just molecules binding to receptors, just physics and chemistry doing what they do. The cytoplasm inside the slime mold flows in rhythmic pulses. It doesn't flow steadily, it pulses back and forth, like a heartbeat, but throughout the entire organism. These pulses coordinate somehow, synchronize, create waves of flow through the slime. When one area encounters food, the pulsing changes, becomes stronger in that direction. The flow increases. More cytoplasm moves toward the food. This strengthens that pathway, makes it more pronounced, makes it thicker. The slime mold is growing toward the food without any brain telling it to. Meanwhile, areas that don't find anything useful start to thin out. The cytoplasm flows away from them, toward areas that are finding resources. It's like water flowing downhill, not intentional, not chosen, just following gradients, following the path of least resistance, following chemistry. But the result looks intentional, looks smart. The slime mold appears to be choosing the best paths, appears to be thinking. But it's all just chemical responses, all just flow, all just patient exploration, guided by simple rules. The slime mold also remembers, sort of, not in the way animals remember. It has no brain to store memories, no neurons to encode experiences, but its structure changes based on what it encounters. If it finds food in one direction, it thickens the pathways in that direction. Those pathways remain thicker for a while, even after the food is gone. The slime mold is more likely to search in that direction again. This isn't memory as we know it, but it's a kind of learned behavior, a physical record of past success, encoded in the thickness of slime channels, in the structure of the organism itself. Scientists have tested whether slime molds can learn patterns. They expose them to regular pulses of adverse conditions. Dry air every 30 minutes. At first, the slime mold just responded to each pulse, contracted, protected itself. But after several pulses, it started anticipating, started contracting before the dry air came. It had learned the pattern, not consciously, not mentally, but physically. Its chemistry had adapted, had started responding to time intervals. Something had changed in its internal rhythms, in its pulsing patterns. It was predicting the future based on the past, with no brain, no nervous system, just slime responding to patterns. This challenges our ideas about intelligence, about what it means to know something. We assume knowledge requires a brain, requires neurons, requires something we recognize as thought. But maybe intelligence is broader than that. Maybe problem solving doesn't require consciousness. Maybe learning doesn't require memory as we understand it. Maybe these things can emerge from simple rules, from chemistry, from the patient exploration of possibilities, from the slow flow of slime following chemical gradients. The slime mold doesn't know it's solving problems, doesn't know it's learning, doesn't know anything at all in the way we know things. It just exists, flows, responds to its environment, and through this simple existence, through this patient response to chemical signals, it does things that look intelligent, that look purposeful, that look designed. But they're not. They're emergent. They arise from the bottom up, from countless small chemical reactions happening simultaneously, from the collective behavior of a blob of slime. There's something peaceful about this, about intelligence without thought, about problem solving without stress. The slime mold never worries about making the wrong choice, never doubts itself, never questions whether it's doing the right thing. It just does what its chemistry tells it to do, flows where the gradients lead, explores patiently, and somehow, through this simple process, it survives, thrives, solves problems that would challenge creatures with brains far more complex than it doesn't have. This is life at its most fundamental. Response without reflection, movement without intention, survival without strategy, just chemistry playing out in slime, just physics determining flow, just patience allowing enough time for solutions to emerge. No rush, no urgency, no stress. Just the slow, steady process of slime doing what slime does, following the simplest rules and finding its way through the world. And these simple rules create remarkable patterns. In 2000, Japanese scientists did something unusual. They took a slime mold, put it in a maze, placed food at the exit. Then they waited. They wanted to see what would happen, whether this brainless blob could navigate the maze, find the exit, solve the puzzle. The slime mold had no way of knowing this was a test, no awareness that it was being observed. It just did what it always does, spread out, explored, searched for food, and what it did amazed the scientists. At first, the slime mold filled the entire maze, every corridor, every turn, every dead end. It was everywhere. A thin film of yellow slime covering every surface. This is normal slime mold behavior. When in a new environment, explore everything. Send tendrils in all directions. See what's out there. It's thorough, patient. There's no skipping areas, no assumptions. Every path gets tested. Every option gets tried. The slime mold doesn't rush this. Can't rush this. It just flows slowly through the space, methodical, complete. Over the next several hours, something changed. The slime mold started retracting from dead ends, pulling back from paths that led nowhere. These paths had no food, no resources, no reason to maintain them. So the slime withdrew, pulled its cytoplasm back, concentrated itself. The maze that had been completely filled with slime started showing empty spaces, corridors where the slime used to be, but wasn't anymore. The slime mold was simplifying, streamlining, focusing on what worked. Eventually, what remained was a single, thick strand. One path through the maze, from the entrance to the food, not just any path, the shortest path, the most direct route, the solution. The slime mold had solved the maze, not through trial and error exactly, more through comprehensive exploration followed by elimination. It tried everything, then kept only what worked, what was efficient, what led to food. The dead ends starved. The successful path got reinforced. What emerged was the answer. The scientists were impressed. but they wondered if it was a fluke. So they tested other mazes, more complex ones. Mazes with multiple possible solutions. The slime mold solved those too, always finding the shortest path. Or, when there were multiple equally short paths, choosing one and sticking with it, never maintaining inefficient routes, never wasting resources on paths that didn't lead to food. It was as if the slime mold understood efficiency, understood optimization, but it didn't. It just followed its simple rules, and those rules led to optimal solutions. Other scientists replicated the experiment, used different mazes, different slime molds, different conditions. The results were consistent. Slime molds solve mazes. They do it slowly, patiently, over hours or days, but they do it. And they do it well. Better than many algorithms. Better than random searching. They find good solutions, efficient solutions, solutions that show a kind of intelligence, even though there's no intelligence there. Just chemistry, just flow, just patient exploration. What's remarkable is that slime molds don't make the same mistakes twice in the same exploration. Once a path proves to be a dead end, the slime mold doesn't send major resources there again. It might test it occasionally, a small tendril checking if anything has changed. But it doesn't commit, doesn't invest heavily in known failures. Or this is learning, real-time adaptation. The physical structure of the organism encodes what works and what doesn't. Thick paths mean success. Thin or absent paths mean failure. The slime mold is its own memory, its own record of exploration. The patience required for this is extraordinary. Imagine exploring a maze where every step takes minutes, where you can't see the whole layout, can't plan ahead, can't think about it. You just slowly extend yourself in all directions. Wait to see what you find, then gradually retract from failures while reinforcing successes. This would test human patience, would frustrate us. We want to know the answer now, want to solve it quickly. But slime molds have no such urgency. They take the time it takes, however long that is, days if necessary. The timeline doesn't matter, only the solution matters. And the solution always comes. Given enough time, given enough patient exploration, the slime mold finds food, finds the way, solves the problem, not because it's smart, but because it's thorough, because it tries everything, because it doesn't give up, doesn't get discouraged, doesn't rush. It just keeps flowing, keeps exploring, keeps following chemical signals, and eventually, inevitably, it succeeds. There's wisdom in this, in the patient, comprehensive approach, in trying everything before deciding what works, in letting time reveal the answer instead of forcing it. Scientists started wondering what else slime molds could solve, what other problems this patient, simple approach might address. If they could solve mazes, what about real-world problems? Problems that required finding efficient networks, connecting multiple points, distributing resources. The answer turned out to be remarkable. The simple slime could solve surprisingly complex problems. In 2010, researchers in Japan tried something creative. They created a map of the Tokyo area using oat flakes. Each oat flake represented a major city in the region. They placed these flakes in the same geographic positions as the real cities. Then, they put a slime mold on the map, centered it on Tokyo, and they waited to see what would happen. What network would the slime mold create to connect all these food sources? Would it make any sense? Would it be efficient? Or would it be random, chaotic? The slime mold did what it always does. It spread out, explored, sent tendrils toward every oat flake, every food source. It found them all, connected to them all. But then it started optimizing, started retracting from inefficient paths, started maintaining only the routes that made sense. That balanced efficiency with redundancy. That connected everything without wasting resources. And what emerged was stunning. The network the slime mold created almost perfectly matched Tokyo's rail system. The actual rail system that humans had spent decades planning, designing, building. The slime mold recreated it in about a day. The major routes were nearly identical. The connections between cities matched. The overall structure was remarkably similar. A brainless blob of slime had independently designed a transportation network that matched one created by teams of engineers, by committees, by extensive planning and modeling. The slime just flowed and found the same solution. This wasn't coincidence. It was optimization. Both the engineers and the slime mold were solving the same problem. How do you connect multiple points efficiently? How do you create a network that minimizes total distance while ensuring redundancy, while avoiding unnecessary complexity? These are engineering problems, complex problems, problems that require algorithms and calculations and planning. Or so we thought. Turns out, patient exploration works too. Turns out, slime flowing according to simple rules finds similar solutions to what human intelligence designs. The researchers tried it with other cities, other transportation networks. The results were consistent. Slime molds create efficient networks, not perfect networks, not identical to human designs, but comparable, competitive, sometimes even slightly better. They naturally balance efficiency with robustness and don't create single points of failure. Don't waste resources on redundant connections that rarely get used. They find practical solutions, working solutions, solutions that emerge from simple rules patiently applied. What makes slime mold networks special is that they're self-optimizing. If you cut one of their connections, they adapt, reroute, find alternative paths. They're resilient, flexible, not brittle like some designed networks. If part fails, the rest adjusts, compensates, finds new ways to maintain function. This is because the network is alive, still flowing, still responding to conditions, still able to change if circumstances change. It's not static, not fixed. It's dynamic, adaptive, patient in its constant adjustment to reality. Engineers started paying attention, started studying slime mold networks, started asking if there were principles they could learn, could apply to real infrastructure, to communications networks, to supply chains, to any system that needs to efficiently connect multiple points. The slime mold doesn't know anything about graph theory, about optimization algorithms, about network topology, but it solves these problems anyway, through chemistry, through flow, through simple rules scaled up, through patience. The networks aren't always the same. Different slime molds create slightly different solutions. This is because the exploration is somewhat random, the exact timing of which tendril finds which food source first, the specific path the cytoplasm happens to flow. These details vary, but the overall structure ends up similar, converges on similar solutions. This is what optimization looks like. Not one perfect answer, but a family of good answers, all in the same neighborhood, all efficient, all working, all arrived at through patient exploration rather than clever design. What's peaceful about this is the lack of struggle. The slime mold doesn't agonize over design decisions, doesn't debate the best approach, doesn't second-guess itself. It just flows, explores, responds to what it finds. And through this simple, patient process, good solutions emerge. Not forced, not designed, not planned, just emerged. From the bottom up, from simple rules, from chemistry doing what chemistry does, from slime being slime. Time is essential here. The optimization takes hours, sometimes days. But the slime mold doesn't care, doesn't worry about deadlines, doesn't rush. It takes the time it takes, and in that time, something beautiful emerges. Something efficient, something that works. Not because anyone planned it, but because the physics of flow, the chemistry of response, the patience of exploration, these things naturally lead toward efficiency, toward solutions, toward networks that make sense. Humans could learn from this, could learn about patience in problem solving, about trying many options instead of committing to one too early, about letting solutions emerge instead of forcing them, about trusting that thorough exploration will reveal answers, about taking the time needed instead of rushing to quick but suboptimal solutions. The slime mold teaches us that intelligence isn't the only path to good answers. That patient, comprehensive exploration works too. Maybe works better sometimes. And this teaching extends beyond networks into our own bodies. You might not realize it, but you're full of slime. Inside you right now is a complex network of mucous membranes. Slime lining your lungs, coating your stomach, protecting your intestines, covering your sinuses. Everywhere that your internal surfaces meet the external world, there's slime. Mucus, a protective barrier made of the same basic components as slug slime. Water, proteins, glycoproteins, creating that characteristic viscous protective layer. Your lungs produce about 1 liter of mucus every day. 1 liter. That's a lot of slime. It coats the airways, the bronchi, the bronchioles, all the branching passages that carry air to the air sacs where oxygen gets absorbed. This mucus is essential. Without it, your lungs would be vulnerable, exposed. Every breath brings in particles, dust, pollen, bacteria, viruses, pollution. All of this gets trapped in the mucus, stuck in the sticky layer, unable to reach the delicate tissue underneath. The mucus isn't just sitting there, it's moving, always moving, slowly flowing upward, toward your throat. This movement is created by cilia, tiny hair-like structures on the cells lining your airways. Millions of them, billions of them, all beating in coordinated waves, like wheat in wind, but microscopic, constant. These waves push the mucus upward, slowly, steadily, patient movement that never stops. This system is called the mucociliary escalator. It's literally an escalator made of slime and cilia, moving particles up and out of your lungs, taking 12 to 24 hours for mucus to travel from deep in your lungs to your throat. That's slow, very slow, about 1 centimeter per hour in some parts of the lung. But it's constant, reliable, always working, always cleaning, always protecting. You never think about it, never notice it unless something goes wrong. But it's there, always, keeping your lungs clear. When the mucus reaches your throat, you swallow it. You don't notice, don't taste it. It just goes down, into your stomach. About 1 liter every day. You've been swallowing your lung mucus your entire life. It's normal, natural, part of how the system works. The stomach acid kills anything trapped in the mucus, any bacteria, any viruses. The mucus gets broken down, digested, recycled, nothing wasted, the whole system elegant in its efficiency. Your stomach has its own mucus layer, different from lung mucus, but similar purpose. Protection. Your stomach acid is incredibly strong. pH of about 2. Strong enough to dissolve metal given time. Strong enough to kill almost anything. But it doesn't dissolve your stomach, doesn't damage the stomach wall, because there's a mucus barrier, a layer of thick, protective slime between the acid and the tissue. This mucus is special, alkaline. It neutralizes any acid that gets through, creates a gradient, very acidic on the inside of the stomach, nearly neutral at the stomach wall. The tissue stays safe. This mucus layer is constantly being refreshed. The cells lining your stomach produce new mucus continuously. The old mucus gets mixed with stomach contents, gets digested along with food, but it's always being replaced, always maintaining that protective barrier. This replacement happens every few days. Your entire stomach lining, including the mucus layer, gets renewed every 3 to 5 days. Constant regeneration, constant protection. You never think about it, but it's happening right now, while you read this, while you sleep, always. If the mucus barrier fails, you get ulcers. The acid reaches the stomach wall, damages tissue, creates pain. This shows how important the mucus is, how essential. Without it, the stomach would digest itself. The acid that breaks down food would break down stomach tissue too. But the slime prevents this. This thin layer of mucus. This barrier made of water and proteins. It stands between you and serious damage. Every moment, every day, protecting you with quiet efficiency. Other parts of your digestive system also have mucus layers. Your intestines, your esophagus, anywhere that needs protection from digestive enzymes or from the contents passing through. The mucus lubricates too, makes everything slide smoothly, makes digestion easier, more efficient, less damaging. It's multi-purpose slime, protecting and facilitating, working constantly in ways you never perceive. Your sinuses produce mucus, your nose, your throat. All of it drains, either out through your nose or down your throat into your stomach. More mucus you swallow without noticing. More protective slime doing its job. Trapping particles. Trapping pathogens. Keeping them from reaching your lungs. From establishing infections. This is your immune system's first line of defense. Not white blood cells. Not antibodies. Slime. Sticky, patient mucus that catches threats before they can cause problems. And this mucus does more than just passively protect. The cilia in your lungs are extraordinary. Each one is tiny, about 6 micrometers long. That's 6 millionths of a meter. Smaller than the width of a hair, much smaller. You could fit hundreds of them across a single hair. But despite their size, they're incredibly important, incredibly active. Each cilium beats about 12 to 15 times per second. All day, all night, your entire life, never stopping, never resting, just constant motion, constant work. Each cell lining your airways has about 200 cilia on its surface, 200 tiny beating hairs, and there are billions of these cells. That means trillions of cilia, all beating, all coordinated, all moving mucus in the same direction, upward, toward your throat. This coordination is remarkable. They don't beat randomly, don't work against each other. They beat in waves, like fans doing the wave in a stadium, but microscopic. And instead of creating noise and excitement, they're creating steady, patient flow, moving slime, clearing your lungs. The beat pattern is specific. The cilium bends forward quickly, pushes the mucus, then returns slowly, recovers its position, ready for the next beat. The fast forward stroke moves the mucus. The slow recovery stroke slides beneath it, doesn't pull it back. This creates net movement, net flow, one direction, upward, beat after beat, hour after hour, day after day. The mucus creeps upward, slowly, imperceptibly, but constantly. You produce about a litre of lung mucus daily. That's a litre that has to move from deep in your lungs to your throat, has to travel through the entire bronchial tree, has to be pushed upward against gravity, and it's all moved by these microscopic cilia, by their patient, coordinated beating. No muscles, no pumps, just tiny hairs working together, creating flow through synchronized motion. It's elegant, efficient, continuous. The mucus layer is actually 2 layers. The bottom layer is thin, watery. The cilia beat in this layer. Their tips reach into the upper layer. The upper layer is thicker, more viscous. This is where particles get trapped, where bacteria stick. The cilia push this thicker layer along, move it upward. The 2-layer system is more efficient than 1 layer would be. The thin bottom layer lets the cilia move freely. The thick top layer traps particles effectively. Together, they create the perfect cleaning system. This system works while you sleep, while you're lying down. Gravity is no longer helping. In fact, it might be working against the cilia depending on your position. But they keep beating, keep moving mucus. The flow might slow down, but it doesn't stop, can't stop. Your lungs need cleaning all the time, not just when you're upright. The cilia adjust, work harder if needed, maintain flow regardless of your body position. They're autonomous, self-regulating, responding to local conditions without any conscious control. When you're sick, when you have a respiratory infection, mucus production increases, sometimes doubles, triples. Your nose runs, your lungs feel full. This is your body ramping up protection, making more slime to trap more pathogens, to dilute toxins, to flush out the infection. The cilia work overtime, beat faster, move more mucus, clear it all out. This is why you cough more when sick, why you have to clear your throat. The mucus is flowing faster, building up faster. The cilia are doing their job, trying to clean an infection, trying to protect you. But if you smoke, the cilia stop working well. Smoke paralyzes them, makes them sluggish, eventually kills them. Without functional cilia, mucus accumulates, doesn't get cleared. This is why smokers cough, why they produce so much phlegm. Their escalator is broken. The mucus has nowhere to go, pools in the lungs, creates problems. The cilia try to regenerate if you quit, can recover some function, but it takes time, patience. The damage from years of smoking can't be undone instantly. The cilia have to regrow, have to relearn coordination, have to rebuild the system. Every day, you swallow about a litre of mucus that came from your lungs. You don't notice. Don't think about it. It just happens. The escalator delivers the mucus to your throat. You swallow reflexively, send it to your stomach. Stomach acid kills anything trapped in it. The protein gets digested. The water gets absorbed. The system works perfectly, silently, constantly, cleaning your lungs, protecting you from infections, from particles, from damage, all through the patient work of microscopic cilia beating in coordinated waves, moving slime upward, always upward, and other creatures have evolved even more remarkable slime defenses. In the deep ocean, there lives a creature that looks like an eel, but isn't. It's called a hagfish. and it has one of the most remarkable defense mechanisms in the animal kingdom. When threatened, when attacked by a predator, it produces slime. Not a little slime. Not a reasonable amount. An absurd amount. One hagfish can turn a 5-gallon bucket of water into slime in seconds. Seconds. So the amount is staggering. The speed is incredible. And the effect on predators is immediate and complete. The slime comes from glands along the hagfish's sides. About 100 glands. When the hagfish is grabbed or bitten, these glands release their contents into the surrounding water. What comes out is a concentrated mixture, mucous proteins, and thread cells. These thread cells are coiled up tight, compressed. When they hit seawater, they expand, uncoil. Each thread is incredibly thin, incredibly long, and incredibly strong. They're made of proteins similar to spider silk, and they tangle, mix with the mucus, create a matrix, a web, and in seconds, the water around the hagfish becomes gel, becomes slime. Thick, stringy, expansive slime. This slime is incredibly effective. It clogs predator gills. A fish that bites a hagfish suddenly can't breathe. Its gills fill with slime, get blocked. Water can't flow through. Oxygen can't be extracted. The predator has to release the hagfish, has to retreat, has to clear its gills or suffocate. The hagfish escapes, swims away, unharmed, leaving behind a cloud of slime, a marker of its presence, a warning to other predators. The volume of slime is what makes this remarkable. One hagfish produces enough slime to fill a bucket. If you put a hagfish in an aquarium and stress it, the entire tank can become gelatinous, viscous, nearly solid with slime. This seems excessive, wasteful, but it works. No predator wants to deal with this. Once they've experienced it, they avoid hagfish. The slime defense is so effective that hagfish have survived for 300 million years, essentially unchanged, still using the same slime, same strategy, because it works, because nothing has found a counter to it. The slime itself is fascinating. It's 99.96% water, almost entirely water. But that tiny fraction that isn't water creates the gel, creates the viscosity. The thread cells expand to 10,000 times their original length. One cell becomes a meter-long fiber, thinner than a hair but incredibly long. Thousands of these fibers tangling together in mucous proteins create the slime, create the defense. It's efficient in materials, but extravagant in effect. After the hagfish escapes, it has a problem. It's covered in its own slime. This would be problematic, could clog its own gills, could prevent breathing. But the hagfish has a solution. It ties itself in a knot, literally ties its body in an overhand knot, then slides the knot down its body, from head to tail. This scrapes off the slime, pulls it away, leaves the hagfish clean, ready to produce more slime if needed. The knot tying serves other purposes too, leverage for feeding, escaping from tight spaces. But slime removal is one important function. The hagfish can regenerate its slime glands. After releasing slime, the glands are empty, depleted, but over days or weeks, they refill, produce new thread cells, new mucous proteins. Get ready for the next attack. The hagfish doesn't waste energy maintaining full glands all the time, only produces slime as needed. As threat levels require, it's efficient, responding to actual danger rather than hypothetical danger, conserving resources until they're truly needed. Scientists study hagfish slime for practical applications, and the threads are incredibly strong, incredibly fine. If we could manufacture similar fibers, they could be used in materials, in fabrics, In composites, spider silk is famous for its strength, but hagfish slime threads are comparable, and hagfish produce much more of them much faster. The challenge is replicating the production, understanding exactly how the hagfish makes these threads, how it stores them so compactly, how it deploys them so rapidly. Nature solved these problems 300 million years ago. We're still figuring them out. The slime also doesn't dissolve easily in water, doesn't wash away quickly. It persists, hangs in the water, gradually disperses but slowly. This extended presence continues deterring predators even after the hagfish is gone. It's a lingering defense, a warning that lasts. Other fish in the area sense the slime, avoid it, learn that something here produces this unpleasant substance. The hagfish doesn't have to fight, doesn't have to be fast or strong or aggressive. It just has to produce slime, massive amounts of effective, predator-stopping slime. And this is enough. This simple strategy has kept the species alive since before dinosaurs existed. There's something almost humorous about it. This ugly, primitive-looking fish, living in deep water, scavenging on dead animals, burrowing into corpses to eat from the inside out. It seems vulnerable, weak, but it has this one trick, this one defense, and it's so effective that nothing can deal with it. Slime, just slime, but so much slime, deployed so quickly, with such effectiveness that it's enough. It's all the defense needed. It's a proof that you don't need speed or strength or weapons. Sometimes you just need the right amount of slime. And while hagfish slime appears instantly, other slime moves at the opposite extreme. In 1927, a professor in Australia named Thomas Parnell set up an experiment. He wanted to demonstrate something simple, that some materials that seem solid are actually liquids, very viscous liquids. Liquids that flow so slowly you can't see the movement. He chose pitch, a black, tar-like substance derived from coal. At room temperature, pitch looks solid, feels solid. You can shatter it with a hammer, but it's not solid. It's liquid. Just incredibly, impossibly viscous liquid. And Parnell wanted to prove it. He heated the pitch until it was soft, poured it into a glass funnel, let it settle for 3 years. 3 years just to let it settle, to let the material distribute evenly, to let any bubbles work their way out. Then, in 1930, he cut the bottom of the funnel, opened a hole, and he waited for the pitch to flow, to drip, to show that it was indeed liquid. And he waited, and waited, and waited. It took 8 years for the first drop to fall. 8 years. The pitch flowed so slowly that you couldn't see it moving, couldn't perceive any change day to day, week to week, even month to month. But it was flowing. Molecules slowly sliding past each other. Gravity slowly pulling the pitch downward. The funnel slowly emptying, just imperceptibly, immeasurably slowly, but moving, always moving, never truly still. The experiment has been running for over 90 years now, since 1930. In that time, only 9 drops have fallen. 9 drops in 94 years. That's about 1 drop per decade. The most recent drop fell in 2014. The next one is expected sometime in the 2020s. No one knows exactly when. The flow is too slow to predict precisely, too variable. Temperature affects it, humidity affects it. Small changes in conditions create small changes in flow rate. And when the flow is this slow, small changes mean years of difference. What's remarkable is the patience required to observe this. Parnell died in 1948. He saw 2 drops fall, just 2. after 18 years of maintaining the experiment. His successor saw drops 3 through 7. The current custodian has been caring for the experiment since 1961. In all that time, through decades of watching, no one has actually seen a drop fall. They happen at night, on weekends, when no one is looking. In 2000, they set up a camera and to catch the next drop, to finally witness it. But the camera malfunctioned. The drop fell in the few minutes the camera was offline. The universe seems to conspire against anyone seeing it. The pitch is estimated to be 100 billion times more viscous than water. 100 billion. That's a number almost impossible to comprehend. It means that in the time it takes a drop of water to fall from a faucet, pitch has barely moved, has barely begun the process. It will take decades to complete what water does in a fraction of a second. This is slime at its most extreme, flow at its most patient, time stretched so thin that years feel like moments. There are other pitch drop experiments around the world. One at Trinity College Dublin, one at the University of Queensland, all showing the same thing, and all demonstrating that what appears solid can be liquid, that stillness can contain motion, that patience reveals truths rushing would miss. These experiments are monuments to slowness, to the value of waiting, to the understanding that some processes simply cannot be hurried. The pitch doesn't care that it's being watched, doesn't care that scientists are waiting, doesn't care about human timescales or human impatience. It just flows, at its own pace, following the laws of physics, responding to gravity and temperature and viscosity, moving because liquids move, flowing because that's what fluids do, just very, very slowly. So slowly that a human lifetime isn't enough to see significant change. So slowly that patience is measured in decades rather than minutes. There's something meditative about this, about a substance that flows but cannot be rushed, about an experiment that requires generations to complete, about patience extended beyond any human capacity for waiting. The pitch teaches us that some things happen on their own schedule. That flow doesn't always mean fast. That liquid doesn't always mean quick. That viscosity can slow movement to nearly nothing, while still allowing it to continue. The pitch is always flowing, right now, this moment, moving imperceptibly, but moving. And while pitch flows slower than almost anything, some creatures are made almost entirely of flowing gel. A jellyfish is barely there. Pick one up, and you're mostly holding water, 95% water. Some species are 98% water. There's almost nothing to them. No bones, no shell, no real structure. Just a thin membrane holding water in a particular shape, holding gel, because that's what a jellyfish is, a bag of gel, living gel, gel that moves, that pulses. That has been pulsing through oceans for over 500 million years. The gel inside a jellyfish is called mesoglia. It's the substance between the inner and outer layers of cells. In some jellyfish, this layer is thin. In others, it's thick, makes up most of the body. The mesoglia is mostly water, but it also contains proteins, collagen fibers, mucopolysaccharides. These create structure, give the gel some firmness, some resistance, keep it from being just liquid, keep it from flowing away. The jellyfish is held together by the viscosity of this gel, by its reluctance to flow. This simplicity is ancient. Jellyfish haven't changed much in 500 million years. They work. Why change something that works? No brain, no blood, no respiratory system, just gel and nerve nets. just the absolute minimum required for life. And it's enough, more than enough. Jellyfish thrive, they bloom, they fill oceans, they outlast more complex creatures. Because complexity isn't always better. Sometimes simple is stronger. The jellyfish moves by pulsing. The bell contracts, pushes water out. This propels the jellyfish forward. Then the bell relaxes, refills with water, ready for another pulse. It's not fast movement, not efficient by fish standards, but it works. It gets the jellyfish where it needs to go, and it costs very little energy. The jellyfish isn't in a hurry, has nowhere urgent to be, just floats, pulses occasionally, follows currents, finds food, survives. Because the jellyfish is mostly water, it's neutrally buoyant, doesn't sink, doesn't need to swim constantly to stay afloat, just exists at whatever depth it's at, pulses gently to adjust position, to stay in water layers rich with food, to avoid getting stranded on beaches. But mostly, it just drifts, lets the ocean move it, accepts where it ends up. No resistance, no fighting against currents, just going with the flow. Literally. Some jellyfish are tiny, smaller than your fingernail. Others are huge. The lion's mane jellyfish can have a bell over 6 feet across, tentacles extending over 100 feet. That's enormous. That's bigger than most whales. And it's 95% water, 100 feet of gel and tentacles, drifting through cold northern seas, pulsing slowly, catching prey, living its simple, patient life. Jellyfish have no brain, but they can sense. They have light-sensitive cells, can tell up from down, bright from dark. They have statocysts, balance organs that tell them their orientation. They have chemical receptors that detect food, detect mates, detect danger. All of this without a central processor. Without neurons organized into anything like a brain, just nerve nets, distributed processing, each part sensing independently, each part responding to local stimuli. When a jellyfish gets damaged, it can often heal. The gel can seal itself, reform, the cells can regenerate. Some jellyfish can even regenerate from fragments. Cut one into pieces, and you might get multiple jellyfish, all clones of the original, all equally viable. This is because there's so little structure to maintain, no complex organs to rebuild, just gel, just membranes, just simple parts that can reform themselves given time. The stinging cells in jellyfish tentacles are remarkable. Nematocysts, tiny capsules containing coiled threads. When triggered, these threads shoot out, harpoon into prey, inject venom, all in a fraction of a second. It's one of the fastest movements in nature, and it's powered by pressure. The thread is coiled so tightly that when released, it explodes outward. All of this happens in gel, in a creature that's 95% water. Precision and power embedded in simplicity. Jellyfish blooms can be enormous. Thousands of jellyfish, millions, all in one area, all pulsing together, drifting together. Why this happens isn't always clear. Food abundance, water temperature, ocean currents. Something triggers rapid reproduction, and suddenly the ocean is thick with jellyfish, thick with gel. Ships have had their cooling systems clogged by jellyfish, had to stop because intakes were blocked by gel bodies. The sheer mass of jellyfish, of water and mucus and simple living gel. It overwhelms mechanical systems designed without accounting for such abundance. The jellyfish will likely outlast us. They've survived every extinction event, every climate change, every ocean chemistry shift. They're adaptable because they're simple, because they need so little, Because gel doesn't require complex systems to maintain. Just water, just basic structure, just the ability to pulse and drift and catch prey. And these abilities have served them for 500 million years, will likely serve them for billions more. Simple, patient, nearly invisible, but incredibly successful. And other creatures use gel in more active ways. An octopus is covered in slime, a thin coating of mucus over its entire body. This isn't dirt, isn't something that needs washing off. It's essential. The mucus protects the skin, keeps it moist, helps regulate what passes through, and most remarkably, it helps the octopus squeeze through impossible spaces, through gaps that seem far too small. The slime is what makes this possible. What makes an octopus able to flow through openings barely wider than its eye. Octopi have no bones, no skeleton. The only hard part of their body is the beak, a small, sharp structure in the center where the arms meet. As long as the beak can fit through an opening, the rest of the octopus can follow. The body is soft, compressible, and the mucus coating lubricates, reduces friction, lets the soft tissue slide through tight spaces, lets the octopus compress itself, flatten itself, elongate itself, whatever shape is needed to fit through. The slime makes it slippery, makes it possible. This ability is remarkable to watch. An octopus approaching a small gap, testing it with an arm, then committing, pushing its body through, squeezing, compressing. The mantle flattens, the arms extend, the whole body becomes elongated, narrow, and it slides through, slowly, patiently. The mucus preventing friction burns, preventing the skin from catching or tearing. Just smooth, slippery passage through impossible spaces. On the other side, the octopus returns to normal shape, expands, resumes its usual proportions, as if nothing remarkable just happened. But the mucus does more than lubricate. It also contains sensors, taste receptors. An octopus tastes with its entire body, with its arms especially. When an octopus touches something, it's simultaneously tasting it. The mucus carries chemical information, delivers it to receptors in the skin. The octopus knows immediately what it's touching. Is it food? Is it rock? Is it another octopus? All of this through touch, through taste, through the mucus-mediated chemical sensing happening constantly across its skin. The arms are covered in suckers. Each sucker can taste independently, can grab independently. An octopus arm cut off from the body will continue functioning for a while, will continue tasting and grabbing, because the nervous system is distributed. Each arm has its own mini-brain, its own processing. The central brain coordinates, but the arms can act semi-independently, can solve problems on their own. And they do this while tasting everything they touch, while sampling the chemical world through their mucus coating. Different species of octopus produce different mucus. Some species coat themselves in thick slime when threatened, make themselves slippery, hard to grasp, hard to hold. A predator grabbing an octopus might find its grip sliding off, might find it can't hold on. The mucus is too slippery, too abundant. The octopus slips away, escapes, leaves behind only slime, only the coating it was willing to sacrifice for freedom. Some octopi can change their mucus chemistry, produce toxins in the slime, make themselves not just slippery, but also unpleasant to touch, irritating, mildly venomous. This is defense layered on defense. First, the slime makes you hard to catch. Then, if caught, it makes you unpleasant to hold. Then, if held, it makes you painful to keep holding. Multiple chances to escape. multiple uses for the same mucus coating. The octopus also uses its mucus to clean itself, to remove debris, to groom. The slime picks up particles. The octopus then wipes it off, scrapes it away with its arms, or lets water flow carry it away. Fresh mucus replaces old, the coating constantly renewed, constantly fresh. This is ongoing maintenance, ongoing production. The mucus glands never stop, always producing, always coating the skin in protective slime. When an octopus sleeps, it still produces mucus, still maintains the coating, because the protection is always needed, the lubrication always necessary. Sleep doesn't stop these functions, doesn't reduce the slime production. The octopus remains coated, remains protected, remains ready to squeeze through spaces or taste the environment. Even in sleep, even at rest, the mucus is constant. Scientists studying octopus mucus have found it remarkably complex. Not just one type of protein, many types, different proteins for different functions, some for lubrication, some for antimicrobial protection, some for chemical sensing, some for attachment when the octopus wants to stick to something. The mucus is tailored, specialized, optimized through millions of years of evolution. It's not simple slime, it's sophisticated slime, multifunctional slime, slime that does exactly what the octopus needs in every situation. And slime serves crucial functions for other creatures too. Frogs are always wet, always slightly slimy to the touch. This isn't dirt, isn't uncleanliness, it's mucus, essential mucus. Mucus that keeps them alive, because frogs breathe through their skin. Not entirely. They have lungs too, but a significant portion of their oxygen comes through the skin, and skin can only absorb oxygen if it's moist, if it's wet. Dry skin doesn't allow gas exchange, doesn't allow breathing, so frogs must stay wet, must stay slimy, or they suffocate. The mucus is produced by glands all over the frog's body, thousands of tiny glands, each one constantly secreting. The mucus spreads across the skin, creates a thin, even coating. This coating never dries out completely. If it starts to dry, the glands produce more, replenish it, maintain the moisture, maintain the ability to breathe. A frog out of water for too long will die, not from dehydration alone, from inability to breathe, from skin too dry to allow oxygen exchange. This mucus does more than enable breathing, it also protects. Amphibian skin is delicate, permeable. Things can pass through it. This is good for breathing, bad for protection. Bacteria can potentially enter. Fungi can attack. The skin is vulnerable. But the mucus contains antimicrobial compounds, peptides that kill bacteria, proteins that inhibit fungal growth. The slime is chemical defense, a barrier that fights pathogens before they can reach the skin itself. Different frog species produce different mucus. Some is mild, some is toxic. Poison dart frogs, those brightly colored species from Central and South America, produce mucus containing potent toxins. Batrachotoxin, one of the most poisonous substances known. A single frog contains enough to kill several humans. The toxin is in the mucus, coating the skin, making the frog untouchable, inedible. Predators learn. Bright colors mean danger. Touching means pain. Eating means death. The mucus protects through chemistry, through poison. But most frogs aren't poisonous. Most just have mildly irritating mucus. Bitter tasting, unpleasant. Enough to discourage predators without killing them. A snake grabs a frog, tastes the mucus, decides it's not worth the effort, releases the frog. The frog escapes. No fight, no injury, just bad taste, just mucus doing its job. protecting through unpleasantness rather than danger. Tree frogs have especially sticky mucus. They need it to climb, to stick to leaves, to cling to branches. Their toe pads are covered in modified mucus, tacky, adhesive, but also releasable. The frog can stick when it wants, release when it wants, move from grip to grip, all through controlling its mucus, making it stickier or less sticky as needed. This is active control, deliberate use of slime for locomotion. When frogs shed their skin, which they do regularly, the old skin is often eaten, consumed. The frog won't waste the nutrients, but before eating it, the frog produces extra mucus, lubricates the old skin, makes it easier to remove, easier to swallow. The process is helped by slime. The frog sits still, the skin loosens, slips off, aided by mucus. The frog pulls it into its mouth with its front legs, swallows it. Nothing wasted. The mucus made it possible. Frogs need to stay in moist environments, not necessarily in water, but in humidity, in dampness, where the air itself contains enough moisture to keep their mucus from drying. Desert frogs exist, but they burrow, hide during the day, come out only at night when humidity is higher, or they secrete extra thick mucus, create a coating that resists drying. Some desert frogs even create a cocoon of dried mucus and shed skin, seal themselves inside it during dry seasons, wait underground for rain. The mucus becomes a survival tool, a way to persist through impossible conditions. The mucus production takes energy, takes resources, water, proteins, compounds for the antimicrobial properties. A frog must eat enough to maintain this constant production, must drink enough to replace the water lost. This is the cost of breathing through skin, of protecting with mucus. But it's worth it. The benefits outweigh the costs. Frogs thrive, spread across the world, live in countless environments, all while covered in slime, all while depending on mucus for survival. When a frog is stressed, mucus production increases. The skin gets slimier, more slippery. This is defense, making the frog harder to hold, harder to swallow if caught. The extra mucus helps it slip from predator jaws, helps it escape. It's an immediate response, automatic. The frog doesn't decide to produce more mucus. Its body just does it. Responds to stress by becoming slimier, by maximizing the protective properties of its coating. And this slimy protection extends even to the smallest communities of life. Every surface in the world is covered in slime. You can't see it usually. It's too thin, too transparent. But it's there, on rocks in streams, on your teeth, on pipes, on leaves, on anything that's been exposed to water and air for more than a few hours. This slime is called biofilm, and it's made by bacteria, and billions of bacteria living together, producing a communal slime coating, building cities of microbes embedded in gel. A biofilm starts with a single bacterium. It lands on a surface, attaches, and immediately begins producing slime, exopolysaccharides, long sugar molecules that create a sticky matrix. This slime helps the bacterium stay attached, protects it from being washed away, creates a foundation. Other bacteria arrive. They stick to the slime, add their own slime to the matrix. Slowly, a community forms, a layer of bacteria embedded in self-produced slime, all living together, all protected by their communal gel. Inside a biofilm, bacteria are different than free-swimming bacteria. They're protected from antibiotics, from disinfectants, from immune cells. The slime shields them, makes chemicals diffuse more slowly, creates barriers. Bacteria in biofilms can be 1,000 times more resistant to antibiotics than the same bacteria floating free. The slime is armor, communal defense, made cooperatively by the residents. Biofilms aren't random. They have structure, channels for water flow, layers of different bacterial species, each occupying a niche, each contributing to the community. The slime holds it all together, creates the architecture, provides the framework on which this microbial city is built. It's organized, functional, a working ecosystem contained in a layer of slime thinner than a hair. The slime on river rocks is biofilm. That slippery feeling when you step on a stone in a stream, that's bacterial slime. Millions of bacteria in a gel matrix. They're not harmful, just living, photosynthesizing bacteria. Bacteria eating nutrients in the water. Bacteria just being bacteria, making the rocks slippery as a side effect of their survival. Creating the biofilm because that's how bacteria live when they can. Together, protected, embedded in slime. Your teeth are covered in biofilm right now. Plaque is biofilm. Bacteria in a matrix of their own making. Streptococcus, actinomyces, other species. All living on tooth surfaces, all producing slime. This biofilm is why you brush your teeth. To physically remove it, to break it up, disrupt it, because the bacteria in plaque produce acid. Acid that damages enamel, that causes cavities. The biofilm protects them while they do this damage, makes them hard to remove, hard to kill. Only mechanical disruption works well. Brushing, flossing, physically breaking up the slime. If you don't brush for a few days, the biofilm thickens, becomes more established, harder to remove. This is maturation, the biofilm growing, becoming more organized, more resistant. Eventually, it can calcify, turn into tartar, stone-like deposits that can only be removed by dental tools. The bacteria created slime. The slime trapped minerals. The minerals hardened. Now it's permanent until scraped off. All from bacteria doing what bacteria do, making slime, living in communities. Biofilms in pipes cause problems. Water pipes, drain pipes, cooling systems. The bacteria attach, build biofilms. The biofilm narrows the pipe, reduces flow, eventually can clog it completely. Cleaning pipes means removing biofilm, breaking up the slime, killing the bacteria. But it's difficult. The biofilm is sticky, attached firmly, resistant to chemicals. It takes mechanical cleaning, scrubbing, or very harsh chemicals. The bacteria don't give up their homes easily, but biofilms aren't always bad. In your gut, bacteria form biofilms on the intestinal lining. These are beneficial biofilms. The bacteria are helpful. They aid digestion, produce vitamins, protect against harmful bacteria. The slime they produce helps them stay in place, helps them resist being washed out, helps them maintain stable populations. This is a biofilm you need, a biofilm that keeps you healthy. Water treatment plants use biofilms intentionally. They create surfaces where beneficial bacteria can grow. These bacteria break down pollutants, clean the water. The biofilm is the treatment mechanism. The slime holds the bacteria in place, keeps them where they're needed, allows them to do their work efficiently. Humans harness biofilms, use bacterial slime for purposes. It's controlled, managed, but it's still biofilm, still bacteria in slime doing what they do. Biofilms are ancient. Among the oldest ecosystems on Earth, bacteria have been forming biofilms for billions of years. Long before animals, long before plants. When the only life was microscopic, it was already forming communities, already producing slime, already building structures from that slime. The strategy works, has worked for longer than almost anything else. simple bacteria, simple slime, complex communities, successful survival. And these ancient communities left evidence that still exists today. Billions of years ago, Earth was a different place. No trees, no grass, no animals, no plants, just rock and water and sky. And in the shallow waters, in the tidal zones, there was slime, bacterial slime, layers of bacteria living together in biofilms, creating mats of microbes, sheets of living slime coating the seafloor. This was among the first life, the first communities, the first ecosystems, made entirely of bacteria and their slime. These bacterial mats still exist, in a few rare places. Shark Bay in Australia, some hot springs, some extremely salty lakes, places where conditions prevent animals from grazing on them, where the bacteria can still form their ancient communities. And in these places, you can see what early Earth looked like, can see the slime mats that covered the planet before anything else evolved. It's a window to the deep past, to the age of slime. The bacteria in these mats photosynthesize, use sunlight to produce energy, produce oxygen as waste. And billions of years ago, these bacterial mats changed the world, changed the atmosphere. Before them, Earth's air had almost no oxygen. After them, after billions of years of bacterial photosynthesis, oxygen accumulated. The atmosphere changed, became breathable, became what we know today, all because of slime, because of bacteria in biofilms producing oxygen as they lived. They didn't mean to change the world. They were just surviving. But their survival transformed the planet. Some of these ancient bacterial mats created structures called stromatolites. These are layered rocks, formed by bacterial mats trapping sediment. Each layer is a mat that got covered in sediment, died, got replaced by a new mat on top, which also got covered. Layer after layer, year after year, million after million, building up stone structures from slime and sediment. These stromatolites can be huge, several feet tall, several feet across, all made from countless thin layers, each layer representing a bacterial mat that once lived, once produced slime, once trapped sediment. Stromatolites are some of the oldest fossils on Earth. Some are over 3 billion years old. 3 billion. That's older than almost everything. Older than animals. Older than plants. Older than complex cells. These rocks are evidence of slime, of bacterial communities that existed when Earth was young, when life was new, when slime mats were the most advanced ecosystems that existed. They're still here, still visible. Proof that slime has been a survival strategy since nearly the beginning of life itself. Modern stromatolites still grow, very slowly, 1 millimeter per year, maybe less. At that rate, building a meter-tall stromatolite takes 1,000 years, at least. Some of the stromatolites in Shark Bay are thousands of years old, living fossils, growing structures that look like ancient ones, working the same way, using the same bacterial processes, the same slime production, the same sediment trapping. Nothing has changed. The method still works, still builds stone from slime, still creates these remarkable structures. Walking on stromatolites, you're walking on living communities, billions of bacteria arranged in layers, all producing slime, all photosynthesizing, all building the structure slowly. Each footstep crushes thousands of years of growth. This is why they're protected now, fenced off, preserved. We almost destroyed them through ignorance, through not understanding what they were. Now we know. Now we protect them. or because they're irreplaceable, their history, their evidence of Earth's youth, of life's simplest successful forms. The slime these bacteria produce hasn't changed. It's the same basic chemistry as modern biofilms, as the plaque on your teeth, as the film on river rocks, water, polysaccharides, proteins, creating sticky matrices, holding communities together, protecting residents, trapping nutrients, trapping sediment, doing what bacterial slime has done for billions of years. Exactly the same. Because it works. Because there's no need to change what already succeeds. These ancient ecosystems teach us about persistence, about success through simplicity. Bacteria and slime have outlasted everything, have survived every extinction, every climate change, every catastrophe, not through complexity, through simplicity, through doing one thing well, making slime, living in communities, working together. These strategies are older than almost anything, and they still work, still allow survival, still create the same structures they created 3 billion years ago. You can visit stromatolites, can see them in Shark Bay, can look at living examples of Earth's earliest ecosystems, can see what covered the planet before anything else. It's humbling. These simple structures, these bacterial communities, this slime, they preceded everything. They made the oxygen we breathe. They created the conditions for everything else to evolve. And they're still here, still slowly growing, still producing slime, still building stone, patient, ancient, enduring. And scientists have even taken some of this ancient slime to space. In 2019, the International Space Station received an unusual shipment. Among the scientific equipment and supplies was a slime mold, Physarum polycephalum, that same yellow blob that solves mazes on Earth. Scientists wanted to know what would happen to it in zero gravity. Would it still move? Would it still solve problems? Would the absence of gravity fundamentally change how slime mold works? They sent several samples, set up experiments, and waited to see what would happen. The slime mold adapted remarkably well. In zero gravity, it still flowed, still explored, still sent out tendrils searching for food. The pulsing that moves cytoplasm around inside it continued, unaffected by weightlessness. The slime mold didn't need gravity to function, didn't need up or down, just followed chemical gradients, just responded to food sources, just did what it always does, but in 3 dimensions now, not constrained to surfaces, able to extend in any direction, able to explore space fully. Without gravity to pull it downward, the slime mold created different shapes, more spherical structures, more even distribution in all directions. On Earth, gravity influences flow, pulls things down, creates preferred directions. In space, there's no preferred direction, no down. The slime mold spread equally in all directions from food sources, created radial networks, symmetrical patterns, beautiful in their uniformity. Different from Earth patterns, but functionally the same. Still efficient, still connecting food sources, still optimizing. The scientists gave it mazes to solve, even in zero gravity, even without up or down. The slime mold solved them, found the shortest paths, so connected the exits, worked exactly as it does on Earth. The lack of gravity changed nothing fundamental about its problem-solving ability, because that ability doesn't rely on gravity, relies on chemistry, on flow, on patience. And those work everywhere, in any environment, under any conditions. The slime mold is universal in its simplicity. Astronauts aboard the station observed the slime mold, watched it grow, watched it move, reported on its behavior. They fed it oat flakes, created experimental setups, all in the strange environment of orbit, where everything floats, where nothing stays put unless attached. The slime mold didn't float away. It stayed attached to its substrate, spread across surfaces, explored methodically, as if gravity had never been there to begin with. This experiment shows something important about slime, about simple organisms. They don't depend on Earth conditions the way complex organisms do. Mammals need gravity, need up and down, need their cardiovascular systems to work with gravity's pull. Plants need gravity to know which way to send roots, which way to send shoots. But slime molds don't care, don't need these cues, just need food, need moisture, need basic chemistry, and those work anywhere, on Earth, in space, probably on other planets. Slime could exist anywhere with the right basic conditions. The students on Earth could control the space slime molds, send commands to the station, set up new experiments, watch via video as their instructions were carried out. It was educational, engaging, showing that science in space isn't just for professional scientists, that students can participate, can learn, can see their experiments carried out in orbit, all through slime, through a simple organism that works the same everywhere. When the slime molds returned to Earth, they were studied, compared to control slime molds that never left. Were there differences? Had space changed them? The results showed minor variations, small differences in growth rate, in exploration patterns, but fundamentally they were the same. Space hadn't broken them, hadn't changed what they were. They remained slime molds, remained functional, remained able to do everything they did before they left. This experiment opens questions about life elsewhere. If slime molds can function in zero gravity, could similar organisms exist in space, on other planets, in conditions very different from Earth? Simple life might be universal, might arise anywhere conditions allow, and slime, viscous flow, biofilm formation, these might be common solutions, might appear wherever life appears, because they work, because they're simple, because chemistry operates the same everywhere. The slime mold didn't know it was in space, didn't care, just lived, just flowed, just solved problems. The environment changed completely, but the organism remained the same. This is the power of simplicity, the strength of simple solutions. They work everywhere, transfer to any condition, adapt without changing. The slime mold is the same in orbit as on Earth, the same floating as grounded, because its success doesn't depend on where it is, only on following simple rules, on being patient, on flowing toward food. And these rules work everywhere in the universe, and humans have found uses for this universal material. As scientists and doctors look at natural slime with increasing interest, not with disgust, with curiosity, with the question, can we use this? Can we learn from billions of years of evolution? Can we apply what slugs and hagfish and bacteria already know? The answer is yes. Slime has medical applications, practical uses, solutions to human problems. Nature figured out slime long ago. We're finally catching up. Slug slime is being studied for surgical adhesives, the mucus that lets slugs stick to wet surfaces in rain. That creates strong bonds, even when everything is slippery. Surgeons need adhesives that work on wet tissue, that work inside the body where everything is moist. Current medical glues often don't work well in these conditions, but slug slime does, has always done. Scientists are analyzing the proteins, the chemistry, trying to create synthetic versions, trying to make glues inspired by slug mucus, glues that could seal wounds, close incisions, hold tissue together while it heals, all because a slug needs to climb a wet leaf. Hagfish slime threads are being studied for their strength. Those protein fibers thinner than spider silk, but comparably strong. If we could manufacture similar fibers, they could be used in sutures, in medical textiles, in wound dressings. The threads are biodegradable, would break down naturally in the body, wouldn't need removal, and they're produced in water, at room temperature. No harsh chemicals, no high energy processes, just proteins self-assembling in water. If we could replicate this, it would revolutionize textile manufacturing, create stronger, lighter materials with minimal environmental impact. Mucus itself is being recreated synthetically. For drug delivery, natural mucus sticks to body surfaces, lines the gut, the lungs, the reproductive system. It stays put, doesn't wash away easily. Drugs embedded in synthetic mucus could be delivered to specific locations, could stay there longer, release slowly, provide sustained treatment. This is already being tested, already showing promise. We're learning from our own bodies, from the mucus we produce, finding ways to use that knowledge to heal. The antimicrobial properties of amphibian mucus are being investigated. Frogs and salamanders produce compounds that kill bacteria, that prevent infections. Some of these compounds are being developed into new antibiotics, new treatments for resistant bacteria. The compounds work differently than conventional antibiotics. Bacteria have less resistance to them. This could be important as antibiotic resistance becomes more common. As bacteria evolve to survive our drugs, frog mucus might provide alternatives, new weapons against infection, all because frogs needed to protect their permeable skin. Biofilms are being studied both as problems and solutions. In medicine, biofilms on medical devices cause infections, on catheters, on implants, on artificial joints. Understanding how bacteria form these slime communities helps us prevent them, helps us design surfaces that resist biofilm formation, or develop treatments that break them down. But biofilms are also being harnessed, used in water treatment, used to produce useful chemicals, used in bioreactors. The same bacterial slime that causes problems can be controlled, directed, used for purposes. Slime molds are inspiring computing, unconventional computing. The way they solve problems without brains suggests alternative approaches to information processing, to optimization, to network design. Some researchers are developing slime mold computers, biological computers that use living slime molds to solve complex problems, to find optimal solutions. It's experimental, unusual, but it works. The slime solves problems that would require significant computer power. Does it patiently, organically, through flow and chemistry, rather than circuits and electricity. The study of viscous flow itself has medical applications. Understanding how mucus moves in lungs, how blood flows in vessels, how cerebrospinal fluid circulates. All of this involves studying viscous fluids, studying slime-like behavior. The physics are complex, but understanding them helps diagnose disease, helps design treatments, helps predict how drugs will move through the body. The same math that describes pitch dripping describes blood flowing, describes mucus clearing from lungs. It's universal, applicable. What's remarkable is how often nature solved these problems first, solved them better, solved them more efficiently than our initial attempts. We invented glues that don't work well on wet surfaces, but slugs had that solved. We struggle with biofilm infections, but bacteria have been forming protective slime communities for billions of years. They're experts, we're students, learning from organisms we once ignored, once considered simple, once dismissed as just slime. Now we know better. Know that simple doesn't mean useless. That slime represents tested solutions, proven strategies, worth studying, worth learning from. And sometimes slime is simply beautiful. Slime can be beautiful. This surprises people. We think of slime as gross, as something to avoid, something unpleasant. But some slime glows, some shimmers, some creates colours that catch the eye, that stop you mid-step, that make you look closer. Beauty exists in unexpected places, and slime, patient slime, holds more beauty than most people realise. Some slime moulds glow in the dark. Bioluminescent slime. In deep forests, in the darkness under logs, certain slime moulds emit light. Soft green light. Faint but visible if your eyes adjust. If you're patient enough to let darkness settle. The light comes from chemical reactions, from compounds in the slime oxidizing, creating photons, creating visible light with no heat. It's the same chemistry fireflies use, the same chemistry some deep sea fish use, but in slime, in a blob of gel creeping across rotting wood, glowing gently, beautifully, in the dark. Slug trails shimmer, that silver line left behind. It's not actually silver, it's mucus, but it catches light, reflects it, creates that pearlescent quality, that shimmer. In morning sun, slug trails look like ribbons of light, like paths drawn in silver. They're evidence of journeys, of slugs moving through the night, and they're beautiful in their way. Delicate lines across concrete, across leaves, across whatever surface the slug crossed. Each one unique, each one a record of patient movement, of slow exploration, of a creature going about its life, leaving beauty as a side effect. Snail slime can create rainbows when light hits it at the right angle. The mucus acts like a prism and separates white light into colours. You see it sometimes on snail shells. On the trail behind them, a flash of colour, red, green, blue. All the colours visible for just a moment, then gone as the angle changes, as you move, as the light shifts. It's fleeting beauty, temporary beauty, but real. The physics of light and viscous fluids creating something that catches the eye. That makes you stop. That makes you appreciate what you would otherwise ignore. Oil slicks on biofilms create iridescence, that rainbow sheen on water, on puddles. Often that's oil from cars, from pollution, but sometimes it's natural oils on bacterial biofilms, on the slime-coating water surfaces. The thin layers create interference patterns, light waves reinforcing and cancelling, creating colours, creating that shimmering effect. It's pollution in urban areas. But in nature, it's beauty, it's chemistry and physics, creating colour from colourless substances. Creating visible light shows from invisible processes. Some frogs have translucent mucus that makes their skin shine, makes them gleam. The slime catches light, reflects it, makes the frog look like it's glowing, like it's wet with something more than mucus. It's just water and proteins. But it's beautiful, especially on bright green frogs, on colourful poison dart frogs. The slime enhances their colours, makes them more vivid, more striking. It's protection and beauty combined, defence and art together. Jellyfish are living art, moving sculptures made of gel, transparent, translucent. pulsing through water. Many are beautiful by any standard, elegant, graceful, their gel bodies moving with fluid motion, their tentacles trailing. Some glow, bioluminescent, creating light in dark water, blues, greens, colours impossible to photograph properly, impossible to capture. You have to see them, moving through water, pulsing, glowing. They're 95% water, barely there, but beautiful, remarkably beautiful. Mucus in sunlight can sparkle. The high points catch light, create tiny bright spots, like stars, like glitter. It's just viscous fluid, just gel, but in the right light, it shines. You see this on morning leaves, on spiderwebs covered in dew and slime, on surfaces where mucus has dried but still holds moisture. Little points of light, thousands of them, creating beauty from something we usually find repulsive, transforming the ordinary into something worth looking at. The colours of slime moulds themselves can be striking. Bright yellow, pure white, deep purple, pink, orange. These organisms have no flowers, no petals, no obvious reason to be colourful, but they are. Bright colours against dark wood, against soil, against leaf litter, standing out, visible, beautiful in their own strange way. Not traditionally beautiful, not flower beautiful, but eye-catching, interesting, worth appreciating. People who study slime often photograph it, share images, show others what they see, what they find beautiful, and those images surprise people, make them reconsider, make them look closer at things they would normally step over, ignore, dismiss. Slime can be beautiful, is beautiful, if you look, if you're patient, if you're willing to see beyond initial reactions, beyond the word slime and what it implies, beyond disgust to appreciation, beyond revulsion to wonder. The beauty is there, quiet, patient, waiting to be seen. And this beauty teaches us something important about patience itself. What does slime teach us? What lesson is there in viscous fluids and patient flow? In substances that move so slowly you can't see them moving? In organisms that solve problems without brains? In survival strategies billions of years old? The lesson is patience, the value of slowness, the power of steady, unhurried movement toward goals. Slime never rushes, can't rush, and yet it succeeds, persists, survives, thrives. Think about the slug, moving at the pace slime allows, never faster, never hurrying. It gets where it needs to go, finds food, finds mates, survives. The slow pace doesn't prevent success, doesn't limit survival. It's just a different pace, a different approach. Not worse, just different. The slug isn't frustrated by its slowness, doesn't wish it could go faster, just moves, steadily, patiently, and that's enough, that's sufficient. Speed isn't necessary for survival. Patience works too. Think about the slime mold, exploring every option, testing every path, taking days to solve problems a computer might solve in seconds, but finding good solutions, reliable solutions, solutions that work. The patience in its exploration pays off. The thoroughness matters. It doesn't skip possibilities, doesn't rush to conclusions, just explores, comprehensively, patiently, until the answer emerges, until the solution becomes clear, not forced, not rushed, just revealed through patient investigation. Think about pitch, flowing so slowly that decades pass between drops. 100 billion times more viscous than water, but still liquid, still flowing, still moving. So never truly still, never truly solid, just patient, impossibly patient. The flow happens whether we watch or not, whether we wait or not, The pitch doesn't care about our timescales, our impatience. It flows at the rate physics allows. And that's enough. That's all it needs. Think about the ancient bacterial mats, building stone from slime, layer by layer, year by year, millimeter by millimeter, creating stromatolites over thousands of years, millions of years. No rush, no hurry, just steady work, steady accumulation, steady building, and the result is enduring, permanent. Structures that last billions of years, built entirely through patience, through consistency, through doing the work slowly but never stopping. We live in a world that values speed, that rewards rushing, that demands immediate results, fast responses, quick solutions, so instant gratification. But slime teaches otherwise, teaches that slow can work, that patient can succeed, that taking time isn't failure, isn't weakness. It's a valid approach, a tested approach, an approach that has worked for billions of years, for countless organisms, for systems and strategies proven through deep time. You can't rush healing, can't force growth, can't shortcut development. These things take the time they take. Like slime, they flow at their own pace. Fighting this doesn't help, doesn't speed things up, often makes things worse. Better to accept the pace, accept the slowness. Trust that movement is happening even when you can't see it. Trust that progress exists even when imperceptible. Like pitch dripping, like slime molds solving mazes, like bacterial mats building stone. The work is happening. See, the flow is continuing. Patience is required, and patience works. Tonight, as you rest, remember slime. Remember the slug moving slowly through the darkness, never rushing, never worried about speed, just moving. Remember the slime mold exploring, testing every option, patient in its thoroughness. Remember pitch flowing, slower than you can perceive, but always flowing, never truly still. Remember that good things don't always come fast, that patience achieves what rushing cannot, that slow, steady movement gets you where you need to go. Eventually, certainly. If you've found peace in these stories of patient slime, if these explorations of slow flow have helped you settle, consider subscribing to Simple Science for Sleep. Tomorrow, we'll explore something else, something new, something equally calming and unexpected. Join us. Be part of this community of people finding rest in learning, finding calm in understanding, finding peace in the quiet corners of science. Please like this video if it helped you relax, and leave a comment telling us where you're watching from, what time it is there, what you learned tonight about patience, about slime, about the value of slowness in a fast world. Let yourself be like slime tonight, not rushing toward sleep, just allowing it, moving toward rest at whatever pace feels natural, not fighting, not forcing, just flowing, like pitch in a funnel, like a slug on a leaf, like a slime mold exploring, slow, patient, steady, trusting that sleep will come, that rest will arrive, that the flow toward peace continues, even when you can't feel it, even when it seems like nothing is happening. You're moving, you're flowing, toward sleep, toward rest, toward the quiet darkness where thoughts slow and slime-like patience takes over, where there's no rush, no hurry, just gentle movement toward stillness, toward peace, toward sleep. Good night.